My name is Deborah Marxsen, and I am presenting the introduction to this AV presentation on nucleic acid delivery for disease therapy. This is a collaborative presentation for Assessment 1 with myself, Cherie, Beahan, and Natasha Purcell making contributions. The subject is Introduction to Biochemistry and Molecular Biology at La Trobe University, and the subject coordinator is Steve Jones. The most important and pressing reason for developing a process using nucleic acids is to fight diseases which have been difficult to effect a cure. For example, viral diseases like AIDS, autoimmune diseases such as rheumatoid arthritis, endocrine disorders, one of which is diabetes 1, hereditary disorders, and cancers. It is a well-known fact that nucleic acids hold genetic information. This information is encoded in the nucleic acids deoxyribonucleic acid, or DNA, and ribonucleic acid, RNA. DNA was first discovered in detail by Friedrich Mischer in 1868. In the 1940s, Oswald Avery, Colin MacLeod, and Malcolm McCarthy discovered that DNA held genetic material. They were able to show that it was indeed DNA, not protein or RNA or other cell characteristics that held the genetic material. In 1952, Alfred Hershey and Martha Chase experimented with bacteriophages. In 1953, James Watson and Francis Crick put all the previous information together and came up with the three-dimensional structure of DNA. After decades more of study carried out by myriad scientists in the fields of biology, physics, and chemistry, and through the laboratory work carried out by Paul Berg, Herbert Boyer, and Stanley Cohen, DNA small portions which were found to be useful were isolated and extracted. And by 1986, a new field was formed, that of genomics. All this groundwork has paved the way towards using nucleic acids in the field of disease therapy. Nucleotides, which are portions of nucleic acids, participate majorly in the metabolism of cells. They work at providing energy and chemically joining cells to hormonal triggers, among other things. The programming in the nucleotides provide the information for the structure of protein and all other cells. And they produce the information for all the functions within a cell. Genes are active coded parts of a DNA molecule which are used in synthesizing biological material and can be synthesized into protein or RNA. With immense amounts of genes inside DNA molecules, cells are generally quite large to accommodate all of the genes. DNA only stores and transmits biological material, which was found with the Ava MacLeod-McCarthy experiment. It was found that DNA, which was taken from one bacterial strain, could be inserted into another bacterial strain, changing the cell and consequently introducing a new sequence of information into the new bacterium. One of the most important reasons for developing a process of using nucleic acids is to fight diseases which have been difficult to treat and to effect cures for as many diseases and disorders as possible. Some methods of nucleic acid disease therapy are gene therapy, transduction using viral vectors, and mechanical transfection using non-viral vectors.
Nucleic acid disease therapy works by introducing genetic material into the host cells to improve cell function, which is causing the disease. DNA crosses the cell membrane using various delivery methods, and the genetic material enters the nucleus, where it utilises the existing cell machinery to undergo transcription into mRNA, which is translated in the cytoplasm to create the necessary proteins to affect the target cell. Single-stranded or double-stranded DNA are introduced to the nucleus and can affect the cell in various ways. Single-stranded DNA can bind to the existing gene, which is contributing to the disease, and prevent transcription of that gene. Some techniques involve the introduction of double-stranded DNA into the nucleus, which can be integrated into the chromosomes, producing proteins that are beneficial to the cell. Other methods involve double-stranded DNA entering the nucleus. However, it isn't integrated into the host cell DNA and is transcribed and translated to produce the beneficial protein. Some diseases, such as cancer treatment, the genetic material which enters the cell produces proteins that cause cell death or increase the cell's susceptibility to treatment. There are two types of gene therapy, those that are applied to somatic cells and those that are applied to germline cells. The use of gene therapy on germline cells is controversial and is not commonly used due to issues with ethics. However, this therapy may one day prevent children being born with debilitating genetic diseases. Nucleic acid delivery therapy can be used for diseases that have a genetic origin. It is also utilised for treatment of cancers and immune disorders and can also be used to improve tissue repair in the body. As mentioned previously, there are various methods for the delivery of nucleic acids to the target tissues. Transduction is the delivery method whereby viruses are used to introduce the genetic material into the cell. The genetic material can then be integrated into the host DNA, or it can be a non-integrating method where the genetic material is not passed on to daughter cells if division occurs. Integrating transduction methods include the use of retroviruses, adeno-associated viral vectors, and poxviruses. The most common retrovirus used was derived from the murine leukemia virus. Retroviruses contain RNA. They enter the cell and the RNA is reverse transcribed into DNA and inserted into the nucleus, where it is integrated into the host cell DNA. A form of retrovirus called lentiviral activates the immune system and can affect both dividing and quiescent cells. Adeno-associated viruses have low immunogenicity, have known pathogenicity, and target non-proliferating cells. An example of adeno-associated viruses is the herpes simplex virus. This is used due to its large size, and it also provides long-lasting effects. HSV-1 replicates within the cell. It causes cell lysis and infects surrounding cells. Poxviruses have large double-stranded DNA genomes. They are often used for immune-based therapy in cancer, and they have no need for a viral vector. Adenoviral vectors are used to introduce DNA into the nucleus, which isn't integrated into the host DNA, making the effects transient. They infect the cell and lose their protein coat in the cytoplasm. The DNA is transferred into the nucleus, where the genetic information is transcribed into mRNA, and the desired protein is translated to form the desired protein in the cytoplasm. Several methods can be used to perform mechanical transfection of cells. Naked DNA can cross the plasma membrane and enter the cell by using electroporation, which increases permeability of the cell, microinjection into the cells, and a gene gun, which introduces the DNA by using small gold particles under high pressure and speed. Liposome-mediated gene transfer is limited by transfection efficiencies in vivo. However, when localized to tumours, pegylated liposomes can increase the effectiveness of chemotherapy. Gene-activated matrix therapy is a method where cDNA is loaded onto a porous biomaterial scaffold and packed directly into a wound. The transfer of genetic information into surrounding cells can be used to improve the repair of tissue.
There are many advantages of nucleic acid delivery for disease therapy. One of those includes specificity. Researchers are able to use nucleic acid delivery for very specific diseases. This allows the therapies to be selective of one particular case and replicate one particular gene line, instead of targeting large areas of a diseased person's body. As a result, every individual can have a disease therapy that targets their own personal case of the disease, instead of one form of medication that can destroy other important cells in the meantime. The biological effects of radiation can result in tissue and organ damage to the body. Radiation is one of the various forms of cancer treatment. In theory, instead of radiation, nucleic acid delivery is able to target the stem cell that is creating the mutated cancerous cells, and instead create healthy cells in the body. This allows specific targeting of the disease cells without sacrificing healthy cells and compromising the health of other body parts. Another incredible benefit of nucleic acid delivery for disease therapy is diversity. Nucleic acid delivery has the ability to cure diverse diseases, as nucleic acid delivery works on the fundamental basis of either inhibition of DNA or RNA expression. Nucleic acids are able to act on a variety of abnormal protein production, whilst leaving other proteins unaffected. Nucleic acid delivery can then work to replace the mutated genes with a copy of the correct gene. As proteins are a fundamental base of many components of the body, nucleic acid delivery is relevant in a wide variety of diseases. In other words, if a genetic mutation is at the basis of the disease, nucleic acid can be considered as a cure. This is exemplified by the 70 different clinical trials taking place specifically on nucleic acid at this current moment. The clinical trials are studying a range of uses for nucleic acid, including malignant glioma, rheumatoid arthritis type 1 diabetes mellitus, bronchial asthma, cystic fibrosis, breast cancer, atopic eczema, Crohn's disease, atherosclerosis, pancreatic cancer, and more. There are very few other treatments that could be considered for so many diverse diseases. Although there are some very obvious benefits of nucleic acid delivery for disease therapy, there are some shortcomings that cannot be ignored. One of those shortcomings includes the cost. The cost of nucleic acid delivery for disease therapy is high. One reason for the high cost is the inability to synthesize nucleic acids for therapy at an affordable price. In turn, this has slowed research considerably. Researching the product is no cheap feat, and this does not include the production and application of the medication. For example, there is a new gene therapy called Luxturna that works to restore vision in patients with inherited blindness. Luxturna treatment until recovery in both eyes is estimated to cost patients between $800,000 to $1 million US. Although there is potential for the cost to decrease as the procedures are approved and financial pressure is put on companies to deliver a more cost-effective option, nucleic acid delivery is not cost-effective. Another shortcoming that must be considered is cellular toxicity. Introducing a high dosage of exogenous nucleic acid to the cells can result in cellular toxicity. Cellular toxicity can result in cellular death, changes in metabolic signaling, and initiate a stress response, all of which are detrimental outcomes. The most common cause of toxicity within the cells is caused by the transfection reagent in the cell in response to transfection. Transfection reagent can be mediated by selecting a transfection reagent that considers the cell type, and thus balances cellular toxicity with high efficiency nucleic acid delivery. Despite this poses an issue for researchers as every cell is different and cell types vary greatly in their sensitivity to delivery and dosage of exogenous nucleic acid and reagent. Another point to consider is the clinical application. The clinical application of nucleic acid for disease therapy poses an issue, as scientists have not yet found a way to compensate for the complex and fragile structure of nucleic acid. Transport and shelf life are two critical components when considering the application of nucleic acid disease therapy in hospitals and outside of the lab. If researchers cannot effectively find a way to transport the fragile therapy, it cannot be used. Despite a few evident shortcomings, there are some very obvious potential and future applications to consider. The potential of nucleic acid delivery for disease therapy is astronomical. The ability to correct a genetic defect is what sets nucleic acid for disease therapy apart from other modern medicines. There are over 70 different clinical trials currently taking place all over the world just focused on nucleic acid therapy. If scientists are able to break down all current barriers, any genetic disorder can be undone. This varies from genetic blindness, sickle cell anemia, prostate cancer, atopic eczema, Crohn's disease, and atherosclerosis to cancers such as cystic fibrosis. The specificity allows scientists to focus on the most obscure and rare genetic disorders and give therapy to an individual case. This also means that diseases that have been incurable in the past would truly be able to be corrected. Crohn's is currently an incurable disease with over 780,000 people in the US affected alone. Crohn's is currently costing the healthcare system between $5,500 and $1,000 for every person every year. Nucleic acid delivery for disease therapy will reduce this cost incomparably and improve the quality of life for hundreds of thousands of people, and this is just Crohn's alone. I like this. There is the potential for delivery of nucleic acids to be simplified and become more cost-effective. Atawala, Han, Kim, and Amiji in their 2017 study discovered the promising future of orally administered nucleic acid therapy. The scientists were able to administer oligonucleotides that could bypass extracellular and intracellular barriers and act upon their target sites within the GI tract. Orally administered therapy provides a sufficient mode of transport and affordable delivery and exemplifies the diversity in which nucleic acid for gene therapy can be used in treatment of disease. These are just a few simple future applications. And like I said, the potential of nucleic acid delivery for disease therapy is astronomical.
Thank you for listening. We have included our references in the following slides if you wish to look into this topic any further.